Welcome to the Deep Dive, the place where we take complicated stacks of research, break them down, and give you the essential nuggets of knowledge you need to be completely up to speed on the world's most fascinating frontiers.
And today, we're talking about a frontier that sounds like, well, pure science fiction, resurrection biology.
Right, the whole field of de-extinction or species revivalism.
Exactly. The process of actively generating an organism that either closely resembles or, incredibly, is an organism that was lost to time.
Okay, so let's unpack this. We've received a really comprehensive set of sources here. They detail the cutting-edge science, the profound ethical debates, all the specific projects that are ongoing, and crucially, the genuine, proven successes happening right now.
It's really not a thought experiment anymore, is it?
Not at all. It's an engineering challenge.
And the science is moving incredibly fast. So our mission for this deep dive is to guide you through the three main technological pathways being used to achieve this revival.
And then weigh the benefits against the very serious ecological and ethical risks.
Right. And then we'll zoom in on the absolutely surprising list of target candidates, which range from the woolly mammoth all the way to ancient date palms.
And as we move through this, there's one critical distinction you need to keep firmly in mind. When we talk about bringing back an extinct species, we're really looking at three major methods.
Cloning, genome editing, and selective breeding.
We're back reading, yeah.
And the tension here really is between genetic purity and, let's call it, ecological function. Only one of those methods, cloning, has the potential to produce an animal with the exact original genetic identity.
The others, genome editing and backbreeding, they're creating something different.
By their very nature, they're creating hybrids or proxies, so they're built to fulfill the role of the extinct species, but they are not the extinct species itself.
That purity versus proxy debate really defines the entire field. The conversation starts now. Let's jump into section 1. The pathways to revival how extinction is reversed. So when you think about resurrection, I think the default image for most people is Jurassic Park. And that relies on the most direct, but also the most fundamentally constrained method, somatic cell nuclear transfer, or SCNT.
What we just call cloning.
Right. So walk us through the technical mechanism here, and maybe more importantly, its massive limitations.
Okay, so SCNT requires a near-perfect starting point. The mechanism involves taking the nucleus.
The genetic blueprint.
The genetic blueprint, exactly. You extract that from a preserved, viable cell of the extinct species. This nucleus is then put into an egg cell that's been denucleated.
Meaning its original DNA has been scooped out?
Carefully removed, yes. And that egg usually comes from the closest living relative.
So you're essentially taking the full instruction manual of, say, the Pyrenean ibex, and you're inserting it into the operational vehicle, the egg cell of a domestic goat.
That analogy holds up on the surface, but the biological reality is just terrifyingly complex.
Yeah.
Because even though the nuclear DNA is from the extinct species,
right,
the resulting organism is still relying on the machinery of that host egg cell. Specifically the mitochondrial DNA, the mtDNA, which governs energy production.
And a mismatch there can be catastrophic.
It can cause profound, sometimes very subtle biological failures.
And that biological failure leads directly to the core constraint you mentioned, the quality of the source material.
Absolutely. SCNT demands well-preserved, viable donor cells, and usually only find those in recently extinct species, things we can cryopreserve or get to shortly after death.
For anything that died thousands of years ago, like a mammoth,
the cellular structure is just too degraded. The DNA might be there in fragments, but the full functional cell is long gone.
We have that one famous, really heartbreaking anecdote that proves both the potential and the profound difficulty of this method.
The Pyrenean ibex. The bucardo.
The bucardo, yeah.
It really is a technological landmark. The last female ibex, her name was Celia, died in 2000. Scientists preserved tissue cultures from her ear.
And in 2003, they did it. They used a denucleated goat egg and a goat surrogate mother, and they successfully achieved the birth of a clone.
It was a moment of pure triumph for resurrection biology.
But the triumph lasted, what, 7 minutes?
Just minutes. The clone died shortly after birth because of severe lung defects. This single fleeting success, it underscores two things for you, the listener.
Okay.
First, the incredible technological feat of SCNT in this context, and second, the profound downstream biological challenges. It just shows that having the DNA isn't enough. if the cellular machinery can't handle that ancient blueprint.
And if we look back at the history of cloning in general, we see similar problems, right? I mean, Dolly the sheep, born in 96.
She had health issues that looked a lot like premature aging.
Exactly. The epigenetic factor, so the way genes are expressed, it seems incredibly fragile when you try to force a regular body cell to, restart the clock of an embryo.
That's the nuance we miss when we just talk about DNA sequencing. Cloning is this delicate operation where the consequences of even minor errors can manifest as severe health defects and short lifespans.
So it's a high-risk, high-reward approach.
And it's really only suited for very recently extinct species where that time gap is minimal.
Okay, so if SCNT needs pristine source material, what happens when the tissue is, say, 40,000 years old, totally degraded and fragmented?
Then we shift completely into the realm of genome editing, specifically using technologies like CRISPR-Cas9.
This is the pathway that's unlocked the kind of iconic ancient species.
It has, because genome editing doesn't rely on a viable extinct cell. It only relies on the information inside that fragmented DNA.
Okay, so for those of us who hear CRISPR-Cas9 and know it's powerful but need the details, the sources mention it started as a bacterial immune system. How does that become our main tool for editing a mammoth?
It's a beautifully elegant adaptation. In bacteria, this system, it defends against viruses. You have a Cas9 protein, which is like a pair of molecular scissors, and it's guided by a little piece of RNA. This guide RNA is programmed to find a very specific sequence of viral DNA. When it finds it, Cas9 cuts it, neutralizes the threat.
So scientists just hijacked that system?
Completely. They simply design a guide RNA to find the target sequence they want to edit, say, the gene for short hair in an elephant, and then they can replace it with the sequence they found in the mammoth genome.
That specific programmability sounds like you've unlocked the cheat codes for evolution. It must be so much faster and more accurate than older methods.
It is transformative.
Yeah.
Because we can now assemble the full genome of extinct organisms from degraded tissues, we can identify those specific adaptive traits.
Which means we can go after a much wider array of species.
Exactly. We don't need a living cell, just the readable blueprint.
So going back to the woolly mammoth example, you can read the genetic code for, let's say, cold-resistant blood, extract that specific sequence, and use CRISPR to splice it into the germ cells of an Asian elephant.
That is the core of George Church's work. They are surgically introducing those key adaptations: long hair, subcutaneous fat, specialized hemoglobin for the cold into the living relative.
But the vital takeaway here is the result is not a pure mammoth.
It is fundamentally a hybrid, or a proxy. You are not reconstructing the full 40,000-year-old genome. You are only making targeted modifications to the modern Asian elephant genome to sort of approximate the extinct species function.
Which means if the resurrected mammoth fails to behave like a true mammoth, if the crucial behavioral genes weren't part of the edit, then the whole premise of ecosystem restoration is undermined.
That is the key insight. The creature you get fulfills a physical role, but it carries the intrinsic behaviors of the elephant. If we miss essential genes for migration or mating or social structure, the proxy might not survive, or it might just fail to do its job.
Okay, let's move to the third pathway, which is less about the lab and more about time and observation. Backbreeding and iterative evolution.
Backbreeding is, you could call it low-tech DeExtinction. It's just artificial selection applied in reverse. You select for ancestral traits that still exist in a population, even if they're rare.
So no fancy genetic reconstruction needed.
Not at all.
This is purely about getting a physical lookalike, a phenotype. The aurochs is the textbook example here.
Correct. The aurochs was this massive wild ancestor of all modern cattle, and it went extinct in 1627. Projects like the Toros program are using selective breeding.
So they're crossing primitive cattle breeds that still have some of those ancient traits?
Right. And they're slowly, iteratively trying to achieve an animal that physically looks and acts a lot like the extinct aurochs.
But while the physical appearance might be restored, the genome is clearly different, and you have to wonder if the behavior is really the same.
Absolutely. The genome will be different, and the new animal might lack the original's full spectrum of behaviors, like its deep instinctual fear of humans. It's a functional mimic, not a genetic revival.
Now, compare that deliberate human intervention to this astonishing phenomenon the sources call nature's de-extinction.
Iterative evolution.
Iterative evolution.
This is where nature just hits the redo button completely on its own. It's when a species goes extinct and then a close relative evolves into an almost physically identical creature to the one that vanished.
The story of the Aldabra rail is perfect for this.
It's one of nature's most compelling miracles. The Aldabra rail was a flightless bird that disappeared about 136,000 years ago when its island was submerged by rising sea levels.
Okay.
Fast forward 36,000 years, sea levels drop, the island resurfaces, and is colonized by a flighted rail. Over time, that new population evolves into a flightless species that's physically identical to the one that perished.
The same bird evolved twice, separated by a global catastrophe.
That's why scientists use the term Elvis taxon. The original is truly gone, and the new one is just a very convincing, ecologically functional cover version.
It's proof that sometimes the ecological niche is just so strong that the same answer emerges over and over.
Right, but it's completely unpredictable. You can't rely on it for urgent conservation.
So finally, before we weigh the pros and cons, let's quickly touch on how these tools are applied to plants and also to species that are only functionally extinct.
Right, plant material generally conserves far better than animal tissue. So scientists can use herbarium specimens that contain viable seeds.
Like the 2,000-year-old date palm we'll get to later.
Exactly. Or they can use something called micropropagation, which is coaxing barely alive embryos back to life. If the seed is viable, the resulting plant is a genetic clone of the historical one. Botanical de-extinction is, weirdly, the easiest path.
And for species suffering from functional extinction, so they're alive, but they can't reproduce on their own.
This is where all the R&D from cloning and genome editing pays off immediately. We use in vitro fertilization, IVF, and artificial insemination to preserve these critically bottlenecked species.
Let's pivot to the rhinos for a second. There are only 2 northern white rhino females left. So how is this de-extinction tech being used to save a species that's still technically alive?
The biorescue team is using preserved sperm from the deceased males, collected long ago, and they're combining it with egg cells collected from the remaining females to create viable embryos.
It's an incredible effort.
It is. The plan is to implant these embryos into surrogate southern white rhinos. This isn't bringing back the dead, but it's using the technology designed for the dead to save the living.
That level of sophistication forces us to ask the big question. If the biological challenge of cloning the ibex led to its immediate death, are we prepared for the much larger ethical and ecological challenge of reintroduction?
That's the core of the debate.
Let's turn now to Section 2, weighing the scale, pros, and cons of reintroduction. We'll start with the upside, scientific and conservation leaps.
The scientific potential is truly immense. I mean, the most immediate benefit is just technological advancement. The R&D needed to sequence an ancient genome or perfect a surrogate mother technique for a mammoth creates breakthroughs that are immediately repurposed for currently endangered species.
That is the most compelling argument for me. If you perfect a complicated embryo transfer process for a silacine hybrid, you can immediately apply that exact same technique to help the critically endangered vaquita or the Sumatran rhino.
Precisely. The genetic technologies advanced for de-extinction act as a kind of, a technological rising tide. It lifts all conservation boats.
And beyond the lab, the optimistic claims are all about ecosystem repair and rewilding. The idea that the returned species can act as a natural machine to reverse environmental damage.
This hypothesis centers almost entirely on the woolly mammoth and its potential role in the Arctic. The argument, heavily championed by George Church, is that reintroducing mammoth-elephant hybrids could fundamentally restructure the tundra ecosystem.
Okay, but this is highly theoretical, right? What is the supposed mechanism for reversing climate damage?
The theory involves 3 main actions. First, the mammoths would graze on dead, insulating grass, which lets spring growth emerge faster.
Okay.
Second, their habit of felling trees and breaking up topsoil would expose the permafrost to more cold winter air, slowing the thaw.
Which would slow the release of trapped greenhouse gases.
That's the theory. So it's not just about bringing back a cool animal. It's about restoring a functional ancient ecosystem, maybe even turning the Arctic back into a grassland.
The audacious goal of the Pleistocene Park Initiative.
Exactly. And on top of that, the sheer charisma of these revived species gives them what's called flagship status.
Ah, the fundraising argument.
Right. Flagship species, like a resurrected dodo, would generate massive public enthusiasm, huge media coverage, and most importantly, money for conservation.
So you could leverage that excitement to raise funds for, say, all of Mauritius's other endangered species.
That's the hope. And finally, there is that powerful ethical argument. Humanity, through our actions, drove species like the dodo and the thylacine to extinction. So we have an ethical obligation to use our science to repair that damage.
That sense of moral accountability is a powerful driver. But this is only one side of the ledger. We have to flip the coin now and look at the downside. the serious ecological and ethical pitfalls.
And this is where it gets really tricky.
I think the most immediate concern for ecologists is the risk of creating a new invasive species.
That risk is very real, and it's often understated. If a species has been gone for centuries, its original ecological niche has probably been filled by something else.
So when you reintroduce the long-gone animal, it could just become hyper-competitive.
It could lead to a scramble for resources and potentially cause the extinction of other existing species.
Or if you bring back a predator, you might just decimate a prey species that hasn't seen that kind of pressure in thousands of years.
The ecosystem is a finely balanced system. Reintroduction carries an enormous risk of destabilizing the whole thing, which leads to the environment mismatch problem.
Meaning if the habitat is changed drastically due to logging or climate change, the reintroduced species may just not survive at all.
Not only that, but they might lack immunity to modern diseases. And the original reasons for their extinction may still be threats. Imagine bringing back the woolly mammoth only for it to be poached again for its ivory.
Right, the environment they'd return to is fundamentally different.
And this feeds into the biggest financial hurdle, the cost and conservation trade-off. We're talking about hundreds of millions of dollars being invested into colossal biosciences and other projects just to bring back a handful of species.
Realistically, that funding competes directly with immediate crisis-level conservation budgets.
So if millions are diverted from programs to save, say, the vaquita, with maybe 8 of them left, we could inadvertently weaken those efforts and accelerate the extinction of species still alive today.
It raises a huge question of prioritization. Should the immediate rescue of species on the very edge take absolute priority over resurrecting the long gone?
And then there's the proxy problem again. If the resurrected animal is only a hybrid, it may not even achieve the environmental benefits that were promised. The immense cost will have been spent for an expensive, genetically modified zoo animal.
The final, and I think the most difficult pitfall, is what the sources call the high cognitive ethical dilemma.
This is where the debate moves entirely out of the animal kingdom.
Yeah. The sources highlight the potential application to species like Neanderthals or other hominids.
And the idea of creating sentient beings without a culture, a family structure, or a safe place in our world
It's truly chilling.
It creates a tremendous burden of responsibility. They would likely have self-awareness and capacities for suffering. If we treat them as experimental assets rather than individuals, that presents a profound, maybe unanswerable ethical challenge.
We just have no legal or moral framework for that.
None. And this is why the global conservation community has recognized the gravity of this. The IUCN's De-Extinction Task Force, formed back in 2014, was a massive early step to try and draft some guiding principles before the science got too far ahead of us.
Okay, let's move from the theoretical challenges to the reality of the lab and the field in Section 3. In the lab and the wild candidates and successes, this is where we see exactly which pathways are being applied to which animals.
And we start with the genome editing focus, the Pleistocene giants and iconic birds.
Of course. The woolly mammoth, as we said, it's the flagship project.
It's the ultimate test case for genome editing and proxy creation, since there are no viable cells for cloning.
So George Church's team in Colossal Biosciences are migrating specific genes into the Asian elephant. What are those precise target traits they're going after?
They're focusing on what you could call the cold climate trifecta. Genes for cold-resistant hemoglobin, for oxygen delivery in freezing temperatures.
Okay.
Genes for dense, long, shaggy hair. And genes controlling the accumulation of thick layers of subcutaneous fat for insulation.
And we just saw a fascinating incremental success on this path.
That's right. In March of 2025, Colossal announced the birth of woolly mice. Now, they were careful to note they mostly used known mouse genetic variants, not pure mammoth DNA.
But it's a proof of concept.
It confirmed the capability to successfully edit and transfer complex traits related to temperature regulation and hair structure. It's a necessary de-risking step before trying the much more complex elephant-mammoth hybrid.
Another major target using a very similar method is the thylacine, the Tasmanian tiger, which died out in 1936.
The thylacine is a fascinating, unique challenge because of marsupial biology. Colossal is partnering with the University of Melbourne. They claim a 99.9% complete genome reconstruction as of October 2024.
And they're targeting its closest living relative, the fat-tailed dunart.
Yes, a small, mouse-sized marsupial.
So what's the specific marsupial challenge here?
Well, marsupial gestation is radically different. The baby is born extremely underdeveloped and finishes its development in a pouch. So the hurdle isn't just creating the hybrid embryo, it's engineering the Dunart to successfully carry, birth, and pouch a much larger thylacine Dunart hybrid.
And they're aiming for a revival attempt as early as 2027.
It's ambitious.
Colossal has also tackled the direwolf, which really showcases how this genome editing creates a sophisticated proxy.
Right, the direwolf project, they analyzed fragmented genomes and determined that the direwolf was much more distinct from the gray wolf than we thought.
So they made targeted edits to the genome of the modern gray wolves to replicate the direwolf's distinctive features.
Exactly, which resulted in the 3 famous pups showcased in April 2025, Romulus, Remus, and Khaleesi.
And this was a proof of concept.
It was designed to show that only minor genetic modifications were sufficient to revive the species' functional characteristics, even without splicing in ancient DNA. It shows how the line between a genetically edited modern species and a proxy is getting very blurry.
We also see iconic birds being targeted, which is especially important for fragile island ecosystems.
The dodo, driven to extinction by humans, is a prime target for Colossal. The goal is restoring biodiversity to Mauritius. Its closest living relative, the Nicobar pigeon, provides the genetic framework.
And the elusive ivory-billed woodpecker.
This bird is considered functionally extinct. There have been no confirmed sightings for decades. Colossal plans to use genome editing on relatives like the pileated woodpecker.
Okay, let's pivot away from the high-tech lab and look at backbreeding in action.
The aurochs restoration is the definitive example here. The Toros program in the Netherlands is actively selecting and crossbreeding primitive cattle breeds
to get an animal that not only looks like the aurochs, but can function as a self-sufficient grazer.
And the objective isn't just historical curiosity, it's ecological. They want to restore its role as a massive keystone species in Europe, maintaining biodiversity by trampling scrub and dispersing seeds.
I saw they're aiming for wild releases in places like the Scottish Highlands by 2026.
It's happening fast. And another great example is the quagga project in South Africa. The quagga was a subspecies of plains zebra, striped only on its face and upper torso.
So they use selective breeding of plains zebras to specifically select for that partially striped look.
Correct. And their goal is immediately functional. They plan to reintroduce these animals to the Western Cape to help eradicate invasive tree species. The quagga proxy is being engineered as a biological tool for habitat management.
Finally, let's look at 2 triumphs in de-extinction, which often involve plants or species that were only functionally extinct.
We revisit the northern white rhinoceros. The biorescue team in Colossal are actively fighting functional extinction. They are generating embryos using cryopreserved sperm and female egg cells. The goal is to implant these into surrogate southern white rhinos before the end of 2024.
It's conservation using the ultimate genetic tools. And then there's the Yangtze giant soft-shell turtle.
A sad, poignant reminder of how quickly functional extinction can become complete extinction.
The last known female died in 2019.
And another one found in 2020 died in 2023. They are still searching for a female to breed with the remaining males, but it just underscores the razor's edge of urgency.
Now let's talk about a biological miracle, the Judean date palm. This is botanical de-extinction at its simplest and most profound.
The Judean date palm was considered extinct around the 15th century. But in 2005, scientists achieved the oldest successful de-extinction by germinating 2,000-year-old preserved seeds.
2,000 years old? How did they even revive them?
It was shockingly low-tech. Simple hydration and soaking, using a household baby bottle warmer to maintain temperature, along with some fertilizer and growth hormones.
Amazing.
The first male plant to grow was named Methuselah. And then in 2021, a female plant named Hannah produced the first dates grown from this ancient lineage in millennia.
That story is such a powerful counterpoint to the complexity of the mammoth. Sometimes the solution is just simple preservation.
It confirms that seed banking is a robust and essential pathway. We saw similar successes with the York groundsel, extinct in the wild by 2000. Seeds from the Millennium Seed Bank were germinated and successfully reintroduced in York in 2023.
And finally, the Floriana giant tortoise, which is a slow, iterative hybrid recovery.
Extinct around 1850, but scientists found living hybrids on another island, so a breeding program is now back-breeding those hybrids generation after generation to recover the Floriana genetic traits.
And they've had success.
As of 2025, over 400 tortoises have been hatched. It's a multi-generational hybrid-based success story.
We've covered a staggering amount of ground here. From dire wolf pups to ancient palms producing fruit again, it really confirms that the science is accelerating beyond prediction.
The capability is no longer the main issue. the resource allocation and the ecological consequences are.
As we wrap up this deep dive, let's take a breath and recap the immense tension we've explored between the promise of restoration and the pitfalls of reintroduction.
We've established that de-extinction offers extraordinary technological tools, tools that could potentially save species on the brink, boost genetic diversity, and lead to massive advancements.
But the investment is huge, often diverting scarce resources from immediate crisis conservation efforts. The risks of ecological disruption are very real.
The global conservation community acknowledges this high-stakes game. The establishment of the IUCN's De-Extinction Task Force back in 2014 was a clear and necessary signal that this technology is being taken seriously.
They're trying to draft guiding principles for its ethical use, recognizing we need a moral compass before we fully embrace this power. So what does this all mean for you, the learner, as you process this monumental shift in our ability to manipulate life and death?
Well, given the simultaneous breakthroughs for functionally extinct species like the rhino and for long-dead species like the mammoth, the question of resource allocation is paramount.
That raises an important question. How should researchers and conservationists balance the immense cost and effort?
Should the immediate rescue of species on the very edge, the ones we know can survive today with aggressive intervention, take absolute priority over resurrecting the long gone? Or do the radical advancements gained from pursuing the most difficult projects, like the mammoth, offer the only long-term hope for saving all life by fundamentally changing the tools we have available? That tension between immediate rescue and future-proofing all life is the defining moral and fiscal challenge of the resurrection biology era.